Hello everyone. So today we are going to learn about endospore staining. So it's a type of staining method that we have learned in our previous video. So today we'll learn about this staining, that is endospore. Okay. So before moving forward, we need to know what endospores actually are. Okay. So we'll have a brief discussion about it. Okay. So endospores are the dormant structures that are formed in various bacteria under unfavorable conditions. Okay, so these are the spores that are formed during unfavorable conditions only. In normal conditions, the spore doesn't form. Okay, and they have very irregular sizes. They are highly refractile and they are heat resistant. And this endospore staining has allowed to differentiate between the spore and the vegetative cell. Okay, so if you see this picture over here, you can see that this is a cell and the cell has a spore inside it. Okay, so this is also one type of cell and this it has spore outside it. Okay, so these green portions are the spores and the pink portions are the vegetative cells. Okay, so if the cell has a spore inside it, it is called endospore, and if the cell has a spore outside it, it's called exospore. Okay, so these spores are actually made inside the vegetative cells, and only in rare cases or in worst cases, the spores comes out of the vegetative cells. Okay, and they form exospores. Now, not all bacteria form spores. Some of the bacteria, mostly anaerobes and some members of bacilli group, they only form these spores, endospores or exospores. Okay, and this is formed during the unfavorable conditions, when the conditions are not right. Now these spores, when they are formed, they are surrounded by many layers, many impervious layers, which are called spore coat, and this coat prevents the core of the spore, which has the important hereditary material like the DNA, they all protect it. Spore coats protect the spore coat. Okay, spore coat protect the spore core. Okay, so we will learn about it in our later slides. So here in the worst situation, you can see the spores comes out from the cell like the exospore, and what they do, they survive independently without the vegetative cell. And when the favorable conditions returns, the spores, what they do, they start germinating. They start germinating on their own and they give rise to a new vegetative cell. So each of the spores has the capacity to build a new vegetative cells and then again come alive. Okay, so that is why the spore forming bacteria are very harmful to humans or any other animals. Okay, the spore forming bacteria are highly pathogenic in nature. Now, coming to the scientists who have discovered this staining technique. This technique was first discovered by Dorner in 1922. Okay, so the actual picture is actually not available. So but it was first discovered by Dorner in 1922. It is a type of differential staining method. So for about the different types of staining methods we have learned in our previous video. If you haven't checked it out, the link is in the description. You can go and check it out. So from there we have learned that there are two types of staining method available. One is the simple staining method and another is the differential staining method. Now this differential staining method, it differentiates between the structures. Okay, so this is what the differential staining method do. So endospore staining is a type of the differential staining method. Now the technique that was proposed by Dorner has a heating process which was very time consuming. So he was kind of unable to define or give a heating method, proper heating method for the endospore. Okay, so in 1933 Another two scientists, they came and they modified his training procedure. They were Schaffer and Fulton. The Schaffer and Fulton, what they did, they gave a proper heating step in the staining method, and they have used primary stain and secondary stain. Uh, Primary stain is malachite green and the secondary stain is safranin as the primary and secondary stain they have taken. Okay, and here in this technique the endospores appear green and vegetative cells appear pink. So this endospore staining is also sometimes referred to as Schaffer-Fulton staining, and this is now commonly known as Schaffer-Fulton staining only. Okay, so they did this modification in 1933. So this is how the cells will appear after the staining. Not all the bacteria take up the color. Take up the endospore color because they don't have the spores inside it. Only the spore forming bacteria will take up the spore color and they will appear as green in color. Okay. So now coming to the requirements that are required for the staining. So first you need a culture of spore forming bacteria. Okay, so some of the examples of spore forming bacteria are Bacillus cereus, Bacillus subtilis or Clostridium botulinum. These are some of the examples you can take. Second is a malachite green, which will act as a primary stain. Then is a safranin, it will act as a secondary stain. 0.5% is used for both of them. Then Bunsen burner is required, staining tray, inoculating needle, water bath and microscope. Okay, so these are the requirements that we need for this endospore staining. Now here in the procedure, if you see in the step 1 we have smear preparation. You have to make the smear first. Then you heat fix it using a spirit lamp. After heat fixing, what you do, you flood the slide with 0.5% malachite green and you keep it in the water bath for 5 minutes. You should remember that the stain should not be boiled. Okay. So you keep it for 5 minutes and after 5 minutes you wash the slide with water. Then in step 6 we have secondary stain. So put the counter stain, that is the safranin, over the slide and keep it for 30 seconds. Now after 30 seconds, wash the slide again, blot it dry and observe under oil immersion microscope. Okay, so this will become more clear if we see this pictorial graph. So here you can see the smear is being prepared. Then what you do, you heat fix it properly. Then you cover the slide with malachite green. So if the bacteria have spores inside it, they will take up the malachite green color, right? Now what you do, you put it in water bath for 5 minutes to allow the stain to enter into the spores. Okay. Then what you do, you wash it with water. Then put safranin over it for 30 seconds again. Then again wash it with water. Then blot it dry and then observe under microscope. Okay. So this is the pictorial graph of this whole staining procedure. It's not difficult. It's quite simple. Okay. It's not that difficult. So you have to just do every step cautiously. Now what we'll see under the microscope. So if we look under the microscope, we'll see that the endospores will stain bright green as they will take up the malachite green color and appear green, and the vegetative cells, they will appear pink as they will take up the safranin color, that is pinkish. Okay. So this is what you will see. So this is a pictorial image of Bacillus subtilis. This is a microscopic image of Bacillus subtilis. Here you can see the endospore. This is the dark green color at one corner of the bacillus. So this is the bacillus. Bacillus in shape also. Okay, so you have these spores at one corner of the bacillus. These are the endospores. Now here is an image provided by ASM Microbe Library. Here you can see the vegetative cells has taken up the pink color of safranin, and the spores have taken up the green color of the malachite green. Now here you can see that not all bacteria have formed the spores. Not all the bacillus have formed the spores. Okay, so this bacteria is actually bacillus in shape. This Bacillus subtilis is a bacillus in shape. Now let us look into another example which is not bacillus in shape. Okay, so here, so this is the image of Clostridium botulinum microscopic image. This image is taken under phase contrast microscope, and here you can see the endospore or the whole cell has a different shape, and the endospore also has a different shape. It is somewhat in badminton shape it. Okay, so this is the endospore of the Clostridium botulinum. Now this is the image provided by microbe library. Here you can see in the Clostridium botulinum, here the green color, the spore has taken the green color, and the pink is the vegetative cell. Okay, so this is how you will look under the microscope. So the green will be the spore and the pink vegetative cell will be pink in color. Now what is the mechanism behind this staining process? Okay, why does it appear? Why does it take this color? Why not the vegetative cell take the green color? Okay, so we'll look into the mechanism of it. Now most vegetative cells, they are stainable, they are easily stainable. Okay, because they do not have that much layer. So most of the vegetative cells, they are stainable with standard aqueous dye solutions, no problem. But endospores, they possess a permeability barrier that prevents the dyes from entering unless the barrier is destroyed by heat, chemicals, ultraviolet light, or mechanical rupture. Okay, so the spores, this endospores, what they have a barrier in their permeability. Their permeability is decreased. Why? Because they have the spore coat. Okay, as we learned in our previous one. So they have a spore coat, and this prevents the dyes from entering into it, entering inside it, unless the barrier is destroyed by heating it or any chemicals are given or ultraviolet or mechanical rupture. The stain will not be able to enter inside the spore. Okay, so in this staining method, in our procedure, we have learned that we are heating it using a water bath, right? So this heating process, heating method or heating step is one of the important step in this procedure. So if we look in the structure of the spore, you can see there is a core in the middle, and this core contains the important uh, part of the cell. So the most important part is the DNA. So it contains the DNA and some of the vital proteins that are required for the survival. Now the core is being protected by many layers, as you can see. Just the immediate membrane next to the core is the inner membrane, and then you can see the germ cell wall, and then you can see cortex, then outer membrane, then the spore coat, and then the exosporium. So there are many layers that are protecting the core. So that is why the barrier needs to be destroyed first in order to stain it. Now this barrier develops as the spore coats are formed to protect the cell's DNA and vital proteins, as I said, in the adverse environmental conditions. Now what are the adverse environmental conditions? That are excessive heating, drying, radiation, low nutrient content, low water content. All these are the adverse environmental conditions that makes the bacteria unable to grow because they are not suitable for the growth. Okay. So these endospores, they are structurally and chemically more complex than the vegetative cell. Obviously, as you can see in the structure, in the picture of the structure, so they are more complex. Now here externally, there are more layers, and as the layers forms, calcium ions increases, and the synthesis of dipicolinic acid, it imparts the trait that makes the endospore chemically unique. So what happens as the layer starts on forming one after another, the amount of the calcium ions increases. the amount of the calcium ions in the spore, it increases, and it allows the synthesis of dipicolonic acid. Now this dipicolonic acid is responsible for this unique behavior, is responsible for the survival in the dormant stage. Okay, so this is what it makes the spore unique in structure. Now endospores are often called metabolically inert. What is metabolically inert? That means they do not carry out any type of metabolism when they are in the dormant stage in order to save the energy. Okay, so they do not carry out any metabolic activity. Only basic activities occur which are required to survive in the dormant stage. Only that much occur, not more than that. Okay. So the low level of the endospore activity is associated with the low water content. Obviously, when the water content is low, the activity will get low because it's one type of adverse conditions only. So this is how the endospore or the spore is able to remain for a longer period of time under unfavorable conditions. It's able to survive in unfavorable conditions, and whenever it will get the favorable conditions, it will come back to its original phase, original stage. Okay, so these spore forming bacteria are very harmful to humans or animals. Okay, so this is what we have in this endospore staining. Hope you have understood the whole endospore staining process. So if you want the detailed notes of this video, you can follow our LinkedIn page. The link is in the description. Also, if you like the video, give it a thumbs up and share and subscribe to our channel for more such microbiology updates.